This talk will focus on cloning. First, we'll discuss some fundamentals regarding cells. Next, we'll talk about what cloning is, and last, we'll talk about what potentially cloning can be used to treat human diseases. This talk will focus specifically on characteristics of eukaryotic cells with respect to humans. In order to understand cloning, we must first understand what a cell is. Cells are the fundamental functional unit of the human body. There are essentially three cell parts relating to cloning. First is the cytoplasm, or the body of the cell. Next is the cell's nucleus, or the control center of the cell. Last is the DNA, or the instruction booklet of the cell. One critical point to understanding cloning is how a single cell, after fertilization, produces the 200 different types of cells in the human body. After the sperm and egg fuse, the cell begins to divide, 2, 4, 8, 16 cells. Remember that every cell contains an identical copy of the DNA, the cell's instruction booklet. At about 5 to 6 days post-fertilization, the ball of cells is now a hollow sphere, where the outside wall of the sphere will become the placenta and the inner cell mass will become the embryo. At this stage, each cell in the inner cell mass can read all of its DNA. However, soon after this stage, the cells begin to decide to become a specific cell type, like a kidney cell or a brain cell. One of the critical ways that this occurs is that they lose the ability to read all of their DNA, and they only read the DNA specifically needed to carry out their function. So a muscle cell reads all of the DNA necessary for a muscle cell to work. Now that we understand how a cell decides to become a certain cell type, we can understand what cloning is. One confusing point in biology is that the word cloning can mean several distinctly different things. For example, in molecular biology, cloning can refer to moving small segments of DNA or genes between bacterial plasmids. This is also called recombinant DNA technology. I'll briefly discuss this more on the next slide. The two other prominent methods which cloning refers to is therapeutic cloning and reproductive cloning. Reproductive cloning refers to using cloning to produce an entirely new organism that is identical to the donor. However, therapeutic cloning is the technology used to produce stem cells for the purpose of studying and treating disease. One brief side note on recombinant DNA technology, which is also referred to as cloning. Plasmids are circular DNA elements which can carry small sets of DNA or genes between different bacterial strains. In molecular biology, plasmids serve as sort of the hammer and nails of the trade, in which researchers are able to move genes from one organism to another to better understand how DNA functions and how cells function. We can use these recombinant plasmids then to engineer specific types of DNA. Back to cloning. Cloning is also referred to as somatic cell nuclear transfer, which was first proposed in 1938. This slide outlines a brief history of cloning, which was heavily debated early on. However, in 1984, researchers used a technique called embryo twinning to lay the foundation for successful somatic cell nuclear transfer. In embryo twinning, it involves separating cells at the 2-cell stage and planting them into separate mothers and essentially producing identical clones. However, in 1997, Scottish researchers were able to successfully clone an animal using somatic cell nuclear transfer. And this is the sheep dolly that we've all heard about. So how does somatic cell nuclear transfer work? An adult cell, which is not a germ cell like a sperm or an egg cell, is used in somatic cell nuclear transfer. The somatic cell's nucleus, the adult cell's nucleus, is removed. They take an egg cell and they remove its nucleus. Then they take the adult's nucleus and put that in the egg cell. They chemically shock it. And remember that in an adult cell, like a kidney cell, it cannot read all of its DNA. When they insert the adult nucleus into the egg cell, they then shock it, essentially allowing reprogramming of the cell, allowing that adult cell to be able to read all of its DNA again. This allows the cell to begin to divide into 2, 4, 8, 16 cells, and it allows it to start forming a blastocyst, which has that inner cell mass, which will develop potentially into a new organism. The Scottish researchers who were able to first successfully demonstrate this technique took a sheep's mammary cell or breast cell. They took out the nucleus. They took an egg cell from another sheep and took out its nucleus. They combined the two and then shocked it. They reimplanted that in a pseudopregnant sheep, which is a sheep that has been treated with hormones to make it think it's pregnant so it will readily accept the implanted blastocyst. And the sheep was allowed to carry the developing embryo to full term, in which case producing an identical clone to the sheep that donated the mammary cell or the breast cell. A note of caution though, on this slide there are some numbers boxed in red, and these indicate the number of successful attempts. So the researchers combined 434 nucleus and eggs together. Of those, only 29 actually started to divide. Of those 29 that started to divide, all were implanted. However, only one produced a viable offspring, essentially giving a success rate of 0.002%, a very, very low number. So why clone? Recall that stem cells are cells which can become any other type of cell in the human body. Stem cells are harvested from the inner cell mass of a blastocyst. Cloning is a way to get stem cells which are genetically identical to the donor, thereby preventing any possible rejection problems. This was first demonstrated in 2004 by South Korean researchers who took human egg cells as well as some granulosa cells, or cells which support the eggs from females. They were able to take the nucleus from those granulosa cells and implant them in the egg cells through somatic cell nuclear transfer, chemically shock it, and essentially get those cells to start dividing. They formed a blastocyst, and then the South Korean researchers took the cells from the inner cell mass, the stem cells, plated them out on the dish, and demonstrated that those cells could become almost every other type of cell in the human body. This was very exciting because it provided the first demonstration that cloning can be used to potentially treat diseases. So the stem cells generated from cloning could be potentially used to replace damaged cells in the human body. They could be used for tissue repair, and potentially through tissue engineering could be used to generate an entire new organ. A potential treatment strategy envisioned by scientists or doctors involve taking cells from a patient who, for example, has undergone a spinal cord injury, cloning those cells, producing embryonic stem cells, then taking those cells and making nerve cells. Those nerve cells can then be transplanted back into the patient to potentially alleviate the damage or potentially cure the damage from the spinal cord injury. This technique provides an inexhaustible supply of materials to treat injury and disease. Up to this point, I've been specifically talking about therapeutic cloning. However, reproductive cloning is using somatic cell nuclear transfer to produce an entire new individual. There are significant risks associated with reproductive cloning. As such, scientists generally oppose reproductive cloning. Some of the risks involve that the cell used to make that individual through reproductive cloning would harbor all of the damage previously accumulated by the donor cell. This has been demonstrated in many of the cloned animals. In addition to the health problems, cloned animals also have a significantly reduced lifespan. The use of reproductive cloning for fertility treatment, as I indicated before, has such a low success rate, it is really not a viable option as opposed to in vitro fertilization, which has a much, much higher success rate. So the sum total is scientists generally oppose reproductive cloning, where therapeutic cloning offers a great potential to generate embryonic stem cells which are genetically identical, in other words, there are no rejection problems, to generate cells in tissues which can be used to treat a wide range of diseases. In closing, therapeutic cloning offers a promising medical treatment by providing a renewable source of stem cells which could potentially be used to treat over 100 million Americans with diseases ranging from diabetes to heart